So as a PI for the last 4.5 years in this center, it is my sincere pleasure to introduce Dr. Christos Papadelis. Dr. Papadelis is the Assistant Vice President of Neuroscience Research here at Cook Children's. Prior to this, he came on in 2019 and was the founding director of the center. Since his time here, he has also held other positions, such as Professor of Research and Bioengineering at UT Arlington and Professor of Pediatrics in the Burnett School of Medicine at Texas Christian University. Christos has more than 20 years of experience in magnetoencephalography and electroencephalography from across the world. This ranges from Greece to Japan, Italy to Boston, and now here, which has set Cook Children's Neuroscience Research apart. His work covers a broad range of studies in neuroscience, clinical neurophysiology, and biomedical engineering. The currency of academia, and you'll hear this from Christos over and over if you know him, is papers and grants. And Christos has over 90 peer-reviewed research articles and $7 million in funding from NIH, foundations, and industry. In fact, Christos, as Dr. Stegall mentioned, ushered in Cook's first R01 from NIH, which is the highest and hardest grant to get. So congratulations to him and Cook. Christos is helping to put neuroscience research on the map in bigger and broader ways. He might not remember this, but years ago I asked him, who are you, what are you? Because we often get these titles from our institution where we got our degree or in the current role that we serve, so for instance, bioengineer or professor. And I was like, I don't think that quite sums him up. I sum myself up as a cognitive neuroscientist, and he says, I'm a translational neuroscientist. And I thought that was the perfect summary, and I'm sure you're gonna see a perfect picture of that now in his talk. Welcome our keynote speaker, Dr. Christos Papadelis.
Thank you, Crystal. So good morning, everybody. First of all, I would like to thank for the invitation. It's a great honor to be here and present my work. Today, I will present the work we're doing together with my research group regarding mapping the human brain in both health and pathology. By myself, when Dr. Scott Perry, he convinced me to leave Boston Children's Hospital and Harvard Medical School and move down here to Texas in order to formulate the program. In Boston, I was the director of the clinical MEG program. I was also an assistant professor of pediatrics. And the beginning, I was just by myself here in Dodson Building, with sharing an office with physician. I would start preparing the transfer of my NIH grants. I brought 3 NIH grants from Boston Children's to here. I would start writing some IRB protocols. I set up the academic network here locally. Actually, the first UTA Cook Children's agreement was done for neuroscience research. And 6 months later, literally, COVID-19 pandemic hit, and then everybody started working from home, especially in research in most institutions. But I decided to take a totally different path, and together with 2 members of the team who were coming here every single day, we're wearing masks and protective goggles. There were no vaccination during this time, and then we start purchasing and testing equipment. A few months later, we started performing the first recordings from children with drug-resistant epilepsy. We start collecting a lot of data and eventually publish this work, but also attracting funding from extramural resources like NINDS and other foundations. So in neuroscience research, we study the human brain, and we use 4 different neuroimaging tools, what's called MEG or magnetoencephalography, the high-density EEG or high-density electroencephalography, the MRI or magnetic resonance imaging, and the transcranial magnetic stimulation or the TMS. And I will say a few words about each of the techniques and how they work. I will start with our favorite one, what's called MEG. So the MEG or magnetoencephalography measures the magnetic activity that's generated by the human brain. It's based on the phenomenon of electromagnetic induction, according to which any kind of change in magnetic field generates an electrical current in a nearby coil. So we actually have thousands of neurons in a focal area of the brain. These neurons, they generate a magnetic field, and this is what we measure with coils which are placed outside of the patient's scalp. On the EEG, we measure the electrical activity of the human brain. So we place electrodes on the child's scalp. And this is a quite unique setup we have here at Cook Children's. This is a unique setup at the international level that allows us to record data from the human brain with more than 550 sensors covering the entire child's head. And for the EEGs, like it's quite child-friendly approach, like there's no need for conductive gels to put on the child's head, as with the conventional EEG. We take this net, we soak it in a saline solution, all these electrodes, they become conductive, and we start performing the recordings. I know that like all of you know what's an MRI, but I think very few people, they know how an MRI actually works. And I will try to explain that really briefly. There's complicated physics behind that. So human body is made out of different tissues that they have different concentration of water. And inside of the water molecules, the atoms, they actually spin around their axons like freely. But when they go inside of a strong magnetic field, usually in the magnitudes of Teslas, with scanners of 1.5, 3 Teslas, even now 9 Teslas. So all these axons, they are aligned towards one specific direction. But when we apply a radio frequency, this equilibrium like is disturbed. So the axons like they tilt by 90 degrees. You will see that here. And when we release this kind of radio frequency, then the atoms here, they send back to the sensor signals. And actually, this is what we measure with the MRI. And the energy they send and the timing it takes, like discriminate the different tissues in the brain. So you can see the MRI as a kind of a professional camera that you can adjust the aperture or the speed of the camera and take different pictures of the brain, what we call sequences. And one of our favorites is what's called diffusion tensor imaging, or DTI. You actually are able to assess different kind of fibers in the brain. You see the microstructures in the brain of the kids. Another technique we work is what's called transcranial magnetic stimulation, or TMS. In this method, like we provide strong electrical currents. These electrical currents, they feed a coil. This coil provides a strong magnetic field in the brain, non-invasively, that you'll see that here. And when we activate this kind of motor cortex here, then these neurons, they send an impulse, as you saw down here, to the periphery, to the electromyography, and this is what we measure in the muscles. By using all these kind of techniques, we examine and study several different pediatric neurological disorders, and we work very closely with several members here in the clinical team. But most of the work we're doing is on kids who suffer from drug-resistant epilepsy, and you will hear a lot about that today. So epilepsy is a very common neurological disorder. It's a very devastating also disorder, like kids, they struggle with seizures, affects like the parents and the family and the patient, of course. It has been estimated that in the United States, there are approximately 500,000 of children that they suffer from epilepsy. And usually the first line of treatment for these patients is through the use of anti-seizure drugs. There's now more than 40 drugs that help us control the seizures. But despite of all these efforts and the medications which are available, up to 1/3 of children with epilepsy, they are unable to control their seizure with drugs. They develop what's called drug-resistant epilepsy. And for these patients, the best available treatment is what's called epilepsy surgery. The goal of the surgery is the complete resection or disconnection of the area in the brain that is indispensable for the generation of the seizures, what we call epileptogenic zone. The efficacy of epilepsy surgery has been significantly improved the last years. It has been estimated that more than 60% of the patients, they become seizure-free at least 5 years after surgery, while the safety of epilepsy surgery has also been significantly improved. There's no need for big craniotomies anymore. Like in the past, they opened the scalp, then they resect this area in order for the child to become seizure-free. Now they open a small hole in the brain, and then they place a probe inside, and with heat, with laser, they burn this area, they deactivate this area where the seizures are initiated. So usually the patients, they go into the hospital, they come to the hospital for a whole week during what we call phase 1. And they undergoing different kind of tests. We examine the symptoms they present during the seizures, MRI, EEG, the MEG, and the PET. And all these kind of neuroimaging techniques, we use them to map several cortical areas in the brain. Among all these cortical areas, the most easy area to assess, it is what's called irritative zone. is the brain area where interictal spikes, they occur in the scalp EEG signals, and we'll see what this means. It's a quite nice biomarker, but lacks some kind of specificity. We'll see that later. And this is like how the spikes, they look like in the EEG and the MEG signal. You see this kind of spiky activity. They appear outside of the background activity in the brain, and this is how we record the data. And the task we have in this kind of case is like to find out and localize the underlying generators inside of the brain which are responsible for this kind of activity. In order to do so, we need to solve two complex mathematical problems. I will definitely not go into details. Like behind this slide there are 40 pages of maths. So what we call inverse problem and forward problem. And by solving these problems, we are simply able to project the activity that we measure outside of the scalp with the MEG and the EEG inside of the brain through this kind of source activation maps. Okay, how we do that? The most easy way of assessing that is what we use is called dipoles. Actually, it's a model. And this model makes an assumption that all observed activity at one specific time point, usually the peak of this interictal spike, can be represented by this kind of dipolar source. And this dipolar source is located inside of the brain. And you can think this dipole as a kind of a tiny battery in the brain that has a positive and a negative pole. And then this kind of like battery, it's somewhere localized in the brain. And we do that again and again. We identify all the spikes, and then ideally, this is the ideal scenario. If all these tiny batteries, what we call dipoles, that in one specific area of the brain, then we know we resect this area, and then the child becomes seizure-free. Okay, how all these things applies in clinical practice. I'm pretty sure you have heard about this case. That was a child who suffered from drug-resistant epilepsy for 16 years. She visited 3 different hospitals before coming here to Cook Children's with no success. Finally, the family, she found Dr. Perry. Scott, he involved my team really heavily in this case. We performed simultaneously MEG and high-density recordings. We identified these interictal spikes. And then we localized this kind of activity. You see these dipolar sources in the focal area of the brain. The clinical team was convinced that maybe the activity was coming from there. They placed invasive electrodes, intracranial electrodes in the brain, and then with laser they ablate this area. The child became seizure-free. She remains seizure-free till now. And one of the most emotional moments and rewarding moments for us, the researchers, is when the parents of the kids, they send us like thank you messages in our, actually, Virginia's mother sent a thank you message to me in my LinkedIn account every single year during the anniversary of the surgery. And that's really rewarding for us. Okay, these kind of models and techniques that we use, they involve heavy math, and I will definitely not go into details. It's a really heavy slide, but I will just to mention to you to be aware of the work we're doing here. So one of my peers, the students like Glikerias Dukopoulos, he used computational methods in order to make an assessment how accurately we can localize this kind of activity with the MEG and the high-density G. So what she found that the MEG is blind to sources that they are located here in the crown of the gyrus, but has better localization accuracy than the EEG, although the EEG was able to detect sources like from all kinds of orientations. So she found that EEG and the MEG, they have complementary information in terms of localization accuracy. Another way of assessing the localization accuracy of these two techniques is by using constructions that they resemble the electromagnetic properties of the human head. And this is a project we ran with one of my PhD students, Said Zahromi. So we developed a phantom, a 3D printed phantom from an MRI of an actual patient. And this kind of phantom is made out of different kind of layers. They have different conductivities to be really realistic. And inside of this phantom, we place dipolar sources and in deep areas of the brain, and then we stimulate these sources with a functional generator. It involves a lot of engineering here, and we measure MEG and high-density G. And by doing that, he found out that like the MEG is able to detect sources deep inside of the brain with a localization error of around 20 millimeters. It's really impressive because like the people believe that MEG is blind to deep sources in the brain. Okay, so as we saw, the MEG and the EEG, they offer complementary information. So we came up with the idea, we published this work back in 2023, that if we combine these solutions into a single solution, then we'll have better localization accuracy. And this is what we found out here with this technique, what we call electromagnetic source imaging. There's no other works in the planet doing that. Like, we are the only group, and some other collaborators in Canada. We found out that this kind of technique offers a localization accuracy of around 8 millimeters, very comparable with invasive recordings. So we're doing really unique work on how to localize this kind of activity. Okay, so when the clinicians, they know now roughly where the seizures they come from, they bring the patients back to the hospital for what's called phase 2. And during the phase 2, they place electrodes inside of the brain with what's called stereotaxic EEG. You see how the electrodes are implanted here. Or they put electrodes directly on the cortex. So they open the scalp and they place electrodes on the cortex, and then they measure the activity. This kind of a more old-fashioned technique is not used now so much. And in this case, we try to assess a different kind of biomarker, slightly different. What we call the seizure onset zone is the area where the seizures they start, but we have to wait for a seizure to occur. And then the patients that come here in the hospital, we are waiting and waiting for the patients to develop a seizure. And how these recordings they look like, just an example, we collect video at the same time. The child is inside of the EMU, and we have implanted the electrodes inside of the brain. And this is what you see, the traces here, and we're waiting for the seizures. And then this may take like hours or weeks, and this is what we found out. Here we are waiting, the seizure onset is in these electrodes, and then you try to identify, to localize where this electrode is inside of the brain, and then you know that this is the location, and you know that you can resect this area and the child eventually to become seizure-free. With one of my PhD students, we came up with the idea. We published this work actually a few weeks ago in Brain Communications, Lorenzo Fabri. We came up with the idea that there's no need to wait for a seizure to occur. We can analyze the first 5 minutes of data and then analyze this data. We'll get all this information with this, what we call interictal biomarkers. Interictal means like between seizures. And by making combinations of this biomarker, we're able to identify this region with high precision and to predict the surgical outcome of the patients. But still, one of the problems we have is that it takes time to analyze this data, and then usually the epileptologists, they sit in front of the screen for hours to identify where's the onset of this activity. So with one of my postdoctoral research fellow, Mariak Pardamian, actually he published this paper yesterday in a very high impact journal. So this is really fresh news. So we came up with the idea of developing an artificial intelligence system that can automatically identify the epileptogenic zone and predict the surgical outcome of the patients with minimal intervention from the clinicians. It's very similar to have a ChatGPT and ask the ChatGPT, which area I should resect in order the child to become seizure-free, and the ChatGPT to respond to you. So this is interictal intracranial EEG data, that's the input, and then he developed this AI-based tool. Mayak, he developed this thing. It looks very cool, but doesn't look like that in the code. And the output of that was the specific region in the brain that should be resected in order the child to become seizure-free. And just to make it a little bit more complicated, this is the actual figure from the publication. That's the input into the system, interictal intracranial EEG recordings. This AI tool gives you two outputs. This is the network that will resect and discriminate from the background activity, and also impressively here identify automatically the spikes in these traces. So it saves a lot of time from the clinicians, and you see how effectively it works. These are the traces here, and then this is the annotations of the spikes from the epileptologist. This is what we call ripples, it's more specific frequency bands. And then you see how the tool automatically identify all this kind of items and depict the areas that are responsible for this kind of activity in the brain. That's the future. Impressively, this kind of algorithm offer a very high sensitivity, really good specificity, and a very high accuracy for identifying this area and predict the surgical outcome of the patients. This is quite impressive. And I'm pretty sure like that this work will receive a lot of attention from the scientific community. Okay, so far we identify what's going on if we have all these dipoles in a focal area of the brain. If we resect this area, then we know that this child will become seizure-free. But in several occasions, like we have several dipoles in a large areas of the brain. Imagine like these tiny batteries are all over the place. And then in this kind of cases, like we don't know exactly what to do. We cannot resect half of the brain in order that the child to become seizure-free. So in these cases, like we involve different kind of tools. It's believed that there's a whole network that is responsible for the generation of the seizure. And we involve what's called functional connectivity. Again, it involves some kind of maths, but I avoid equations here. So the functional connectivity actually quantifies the temporal coincidence of spatial distance neurophysiological events, and we see how this applies to a pair of signals from these intracranial recordings, invasive recordings between the blue signal and the red one. We get one value. This value ranges from 0 to 1. 0 is like almost minimal functional connectivity. 1 means maximum functional connectivity. And by doing this kind of comparisons and estimations between all pairs, then we estimate what's called functional connectivity matrix. And then these voxels here, or pixels actually with high values, they indicate what we call critical nodes in the hub. And the critical nodes, they are actually nodes in the area which are highly or densely connected to the rest of the network, and they play critical role of when the network is damaged. So there's no need now to identify a specific region, epileptogenic area. Then you identify some hubs in the brain. With a laser ablation, you burn these nodes, and then eventually the whole brain comes back to an equilibrium, and there's no seizures. Just to make it more simple, it's very similar to when you have a traffic jam here in DFW area. I'm pretty sure like several people like who come here like in the morning from Dallas, for example, or like far away, they struggle with that. And in several of the cases, like there's probably these critical intersections, there's constructions there or an accident. And we know that like if this problem was solved, then the whole network will have less traffic. So by using this concept, like one of my postdoctoral research fellow, Ludovica Corona, she published this high impact paper in Brain. She applied this kind of method with both invasive and non-invasive recordings. This is the MEG and high density G. This is intracranial EEG. And she estimated these functional connectivity maps. And what she found that with all the three methods, this invasive and this is MEG and high density G, she found increased functional connectivity inside of the area that was resected in the good outcome patients compared to outside, an indication that this is a good biomarker for helping these kids. And let's see how this applies in two patients. This is a patient with good outcome. Good outcome is like they resect this area and the child became seizure-free. And poor outcome patients means that neurosurgeons, they resect this area, the child was not seizure-free. So in the first case, we see here with green color the resection. And in the good outcome patient, all the nodes with high values of functional connectivity were inside of this resection. And this area was resected. The child is seizure-free. But in the poor outcome patient, these high functional connectivity nodes, they were outside of the resection, and the patient was not seizure-free. Okay. We have done extensive work on how to identify which area to resect in order for the children to become seizure-free. There's also another important information in epilepsy surgery. We need to identify the brain areas that we should not resect. And that's really important because you can resect one area, you can ablate one area, and then the patient to become seizure-free or will not be able to talk again, or walk again, or move his hands again. So it's important to map what's called eloquent brain areas. There are different areas in the brain responsible for the normal functions of the human body. And how we do that? The conventional way is what's called functional MRI. The kids, they come here in the hospital, they go inside of this scanner, and then we have like, it's a quite tedious process, and most of the tasks they involve there is like quite child unfriendly. For example, for mapping the motor cortex, we ask the child to move his fingers like more than 100 times. So it's really boring. And the child's like, it's difficult to cooperate with that. And that's why the results of this kind of test, they are quite ambiguous. And to solve this problem, we came up with a new concept or idea with one of my postdoctoral research fellows, Yanis. He developed a video game to make it more child-friendly, not in the MRI now, but like in the MEG. So the child sits comfortably in an armchair and plays this video game that involves different kind of components. Auditory, you cannot hear that, but they have like sounds, language. You see there's some words here. They're coming. Visual components, sorry, the video game was stopped. And then a motor, they press a button in order for the child to play the game. And then this very preliminary data, we show here that how we can map this activity nicely in the brain of the kids by just playing a video game. Okay, that was about drug-resistant epilepsy. A significant portion of the kids with epilepsy, they have an underlying genetic component. So actually it has been estimated approximately 30% of the kids, like they have some kind of mutation in their gene that cause epilepsy. And we do quite extensive work with one of these pediatric genetic syndromes, what's called Dravet syndrome. Kids with Dravet syndrome, they come in the clinic, usually the first months of life, they suffer from febrile seizures. Means that the seizures are initiated by fever. Then later they develop other types of seizures. They have present significant developmental delays, difficulties with sleep, growth, and nutrition issues. And the underlying etiology for this kind of syndrome is a mutation in the SCN1A gene. This gene encodes the AE1 subunit of the voltage-gated sodium channel. And what actually this kind of like mutation causes in the brain, it changes the balance between inhibition and excitation in the brain. So in the normal brain, the normal central nervous system, we have two neurotransmitters, what's called the glutamate and the GABA. The glutamate is responsible for the excitation, and the GABA is responsible for the inhibition. It's like you're driving a car, you have accelerator and brake. And then in the case of Dravet kids, like the brake is broken because the GABA is low. So that's why also we see seizures in these kids. And there's now new treatments. That's why this project is funded by a pharmaceutical company, Encode Therapeutics. But the problem with the treatments, like we provide the treatments to the kids, and then we're waiting to see if the treatments, they work. So usually the patients, they keep notes, whether they see less frequent seizures in the child, or if they see improvements in the cognition. So with my team, we came up with the idea to develop a non-invasive biomarker to assess this level of excitation inhibition in the brain. This is a project we ran with one of my PhD students, like Sakar Rijal. We came up with the idea of if we present different kind of stimuli to the kids, visual stimuli, auditory, and tactile stimuli with the MEG and the high-density EEG, we're able to record responses in the brain, what's called beta and gamma activity, and be able to identify this level of excitation inhibition in the brain. I will try not to go into details, but I will just briefly explain that like in kids with Dravet, what we observed that like this component was absent. This is called time frequency analysis plots. At 0, we provide the stimulus onset. We provide a picture, and then we see a few milliseconds later this activity in different frequency bands in the brain, in the gamma and the beta, and the kids with Dravet, this component was absent compared to the healthy controls. But much more impressive was the fact that like this kind of like changes, the non-invasive measurements in the brain, they were correlated with the levels of the GABA in the blood. So we were able, by using this kind of technique, to provide an indirect tool, a non-invasive method for assessing the level of excitation inhibition in the brain. Okay, let me change gears now. Like another disorder that we're doing extensive work with my group is what's called cerebral palsy. Kids with cerebral palsy, they have significant motor deficits in both upper and lower extremities. Half of the kids with cerebral palsy, they need some kind of walk aid in order to be able to walk. And most of the kids, they have also problems with their hands function. And cerebral palsy is kind of an umbrella term. It describes a group of disorders of the development of movement and posture. It's a very common disorder, like actually it's the most common motor disability in childhood. It has a prevalence of 2 per 1,000 live births. And usually the underlying etiology is a kind of a brain insult that takes place either in utero during pregnancy or after birth, and it causes all these problems. let's see how the brain develops in utero. This is like I like this graph a lot because it gives you a good idea of the whole developmental stages that the neurons they take place in utero and after birth. So during first trimester in utero, we have neural proliferation and migration. Then during the second trimester, we have like the development of the white matter in the brain. And then eventually during the third trimester, we have a maturation of the cortex and what's called synaptic pruning. So there's like a selective way of keep specific synapses in the brain. And then usually after birth, we have the myelination of the tracts and the growth of the gray matter volume. And at which timing of this developmental trajectory, the insult, the injury will take place, provides actually very important information about the functional outcome. So for example, if the insult takes place during the first trimester in utero, we got malformations of the basic neural architecture. In the second trimester, periventricular lesions and damage to the white matter tracts. And then in the third trimester, cortical, subcortical lesions. This is like, for example, this is what is called stroke, damage to cortical and subcortical gray matter. And let's see how brain plasticity works in kids with cerebral palsy. This is the development, normal development, healthy child. What we see here with blue colors is motor fibers. The motor fibers, they control upper and lower extremities, how we walk, how we move our hands. And then during birth, these fibers, they project bilaterally. So both left and right hemisphere, they control left and right hands. But as time is passing, especially during the first months of life, then we have a gradual pruning of the ipsilateral side. So eventually we have like one hand or one leg controlled from the other side. The right hand is controlled from the left hemisphere. And at the beginning, that's not the case. That's why when you see the infants, when they were born, they move the hands like all together. They don't have discriminate left and right because like the brain like controls both sides. So let's see now what's going on when we have an insult. For example, this is a case with a stroke, necrotic lesion in the brain. So in this kind of case, the human brain, like the whole process is disrupted. There's damage in these motor fibers from this side. And in order the brain to compensate for this kind of loss, it keeps some of these otherwise pruned fibers to control the parietic hand. For example, you have a parietic hand that controls from both hemispheres, left and right. Left and right controls the right hand. And because of that, what we see this kind of awkward behaviors. This is what we call mirror movements. It's fascinating. You see that the child is manipulating this object, and if you see, the child is doing exactly the same thing, unconsciously, with the other hand. And this is because like one hemisphere controls both hands. And then, I don't know if you can see that, like this is from my lab in Boston. You touch the child's fingers with these monofilaments, and the child does not recognize which finger you touch. There's also sensory issues there. So with one of my postdoctoral research fellow, with Sadras Akhtadian, we do extensive work by using neuroimaging techniques to make mapping of all this activity. With TMS, he mapped these areas in the brain where the motor cortex is located. And then he used this technique I described in the morning, like early in the beginning, like about this diffusion tensor image. It gives these fibers in the brain that they control the upper and lower extremities. And what you see here, we stimulate here the healthy hemisphere. This is the hemisphere with stroke. And then we measure activity, myographic activity to both hands. This means that, like what I told you before, like you stimulate here and then you control both hands. It's not only important to understand the brain plasticity in these kids. It's very important also to see what kind of treatment we are offered to these kids. And this is a project that is funded by NIH. We have this robotic system. It's called Amadeo. The kids, they play actually a video game. They control with their fingers this video game. And at the same time, we measure what's called high-density EEG. You see these traces here. And the kids, they come usually for 15 days here in our research center. We have families, they come in from Dallas, like they were coming every single day for 15 days to receive the treatment. And we make an assessment of motor functions and somatosensory functions before and after to see if the treatment works. We have quite impressive findings. This is like data from 6 kids so far. We see that in most of the kids, we saw significant improvement in the grip force and the range of motion. So most of the kids, like this, we observed objectively that they improved their motor functions in only 15 days with this intensive training. Quite impressively, I asked Sadra, who's running this project with me, to ask the parents of the kids in a subjective way if they observed improvements in the kids through a survey. And most of them, they testified that they observed subjective improvements of the kids' functions of upper extremities. Okay, a few words about that. I will not go into details, just to give you an idea. We collect high-density G at the same time in order to map patterns of brain plasticity, and this is important because we need to understand how the brain is organized as a result of this treatment, and I will definitely go into details like this, a very preliminary data so far. Okay, we saw that with the TMS we are able to map the primary motor cortex, but TMS can be used also for other reasons, and this is a project we run with one of my PhD students, Behnam Gabel. So by applying now this strong magnetic field, but several times, then what we're able to do, we're able to excite the motor cortex here and provide higher levels of excitability in the primary motor cortex. And then by doing that, we hope that kids with cerebral palsy, they will have better outcomes. We submit an IRB protocol. We're still waiting approval for that. And I hope we'll have this kind of research study approved soon. Just to give you another idea about some cool projects I like. Again, this is a project that Behnam he's working on. Some of the kids with cerebral palsy, they suffer from what's called dystonia. The dystonic kids, they have problems the problem is in the deep areas of the brain, like here in the basal ganglia. So Behnam, he developed this kind of cool tool. He put electrodes on the child's scalp, and he provides different kind of stimulation, different frequencies. These frequencies, they cancel to each other, and then they target deep areas of the brain non-invasively in order to provide treatment to the kids. So that's upcoming thing. Okay, I will describe two more projects, and I hope you are not tired. So we do also work in the NICU, in neonatal intensive care unit, with infants. And one of the issues that the neonatologists, they deal with infants in the NICU is that several of these kids, they have this brain insult, brain injury. And eventually they will develop cerebral palsy. Some of the kids, they will develop cerebral palsy. So the problem they have now, in order to diagnose this kind of brain injury, they transfer the kids outside of the NICU to the MRI. They provide sedation. And then this is so these kids are really fragile. And with my team, we came up with the idea whether we're able to develop a non-invasive biomarker at the bedside that can assess the neurological status of the infant. And we came up with the idea of nutritive sucking. So when kids were born, the first thing they would like to do is drink milk, okay, or get nutrition in order to grow. And this is a project we ran with one of my postdoctoral research fellow, Mosde Ascari. That was based on previous studies that have shown a strong relationship between the nutritive sucking patterns and the neurological outcome of the infants which are hospitalized in the NICU. So the sucking, nutritive sucking is controlled by a network in the brain. And for this project, we use this kind of tool. It's an FDA-approved tool. It's called Enfant. So this tool actually measures the movements of the tongue while the child get the milk. And we transfer with Bluetooth the data to a tablet, and we further analyze the data. So this is data we have so far. This is a healthy child. And you see how nutritive sucking movements, they are really smooth, upside down, in a healthy brain, okay? We quantify that with what called smoothness. It's really smooth. And child with hypoxic ischemic encephalopathy is a type of brain injury. You see how these sucking patterns, they are totally disrupted. And this is a project we're still working. We have data we got from Michigan University through a collaboration. This is data from healthy kids with the green color here. You see this level of the smoothness. And kids with HIE, it's right here. These kids from Cook Children's. And you see that during the first month, we have significant differences in the level of the smoothness, an indication that this is a kind of a biomarker that we can help us assess the neurological status of the infant in the NICU. One more idea we're exploring, and this is again the beginning. In our NICU here, we collect high-density EEG it responds to different kind of stimuli. If you see the finger here of the child, we provide tactile stimuli with pneumatic stimulation, compressed air puffs, pop, 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 and then we measure the responses. This is an auditory. You can hear that. We provide auditory stimuli, and then we collect the data from the brain responses, and a visual stimulation. I don't know if you can see that. We provide like flashes to the child, and maybe this. Oh, sorry. Yeah, we provide flashes to the child, and then we measure the response of the child. Preliminary data so far, one healthy child, and then 2 children with hypoxic ischemic encephalopathy. This is the timing. At 0, we provide the stimulus, and then a few milliseconds later, we see the first cortical response, a significant delay in the kids with brain injury, an indication that we have like a kind of a potential biomarker. Okay, last project for today. We do some work on autism spectrum disorder. That's a very new project. I'm not expert on this field, but like I try to study how all the brain works, and we're learning. So kids with autism spectrum disorder, they suffer from different kind of deficits. They have information processing deficits. They have deficits in how they process the emotions. And most importantly, they also have sensory processing deficits. For example, when they hear like really loud sounds, they cannot tolerate them. They close their ears. Or when they see a kind of bright light, they cannot tolerate this kind of external stimuli. So with one of my postdoctoral research fellow, with Yannis Doumanis, we try to examine this kind of process in the brain. We collect data from healthy controls and 5 kids with autism so far. We present different kind of visual stimuli in the MEG, and we measure the brain responses. This kind of stimuli, they have different emotional content, smiley faces, angry faces, other kind of pleasant and unpleasant pictures. And then we found out that the kids with autism, they have higher level of visual sensitivity. They could not tolerate this like kind of visual stimuli. But most importantly, we measure that in the brain. So we measure the red color here is like the response of the kids with autism compared to the healthy controls, a much more higher response to the visual stimuli for the kids with autism, and a significant delay here in what's called fusiform face area. It's an area that's responsible for the face processing of the kids. Still ongoing work here, and that's the science for today. And thank you for your attention, because I know that was a heavy part. And I will close my talk because I would like to summarize like what I have achieved all these years. And when I was preparing the slides the last 2 weeks, I said like, oh my God, like we've done so many things, I don't realize it because like I'm always this hyper excited. And then when you sit down in a more laid back way, it's like, oh, how is it possible to have done all these things in 5 years? And first of all, like, we published 4 scientific publications in top tier scientific journals. And I know that like I hope scientific publications like they count because it adds visibility to the net to the system here. And in 3 of these publications, like we are the leading institution, so adds great visibility. We had 103 papers in peer-reviewed scientific journals. In most of these papers, we are a leading institution. We don't just participate somewhere. We are doing the work. The research is done here at Cook Children's. We have 1 submitted US patent, and we'll have more coming. We established collaboration with academic institutions, like here in the region of DFW, with UTA, of course, with TCU, with the School of Medicine in Fort Worth, Boston Children's Hospital, my previous institution, and MGH, I have close collaborators there, and also internationally, with Campus Biomedico in Italy, and Finland with Aalto University, we have collaboration for the TMS. All these achievements, they attract attention from media. I was for the first time in my life in American TV. And we were in the local newspapers, also like in several like social media, and our work have attract attention in all over the place. Several awards, several invited talks. I don't even remember how many stuff we have done. So very prestigious awards, really difficult to get. To get one of these awards, like you select your abstract 1 out of 1,500 abstracts. So the team is traveling in different places, present their work, and bring the the news that like Cook Children's is doing really amazing work in research. That was the last count when I was counting how many abstracts. In most of them were leading institution again. We brought the scientific community here in Fort Worth. We organized the MEG Texas Consortium meeting that now became national. The next MEG Texas Consortium is not MEG Texas now, sadly, but it's national MEG Consortium. It will be in Florida this April. Covers in scientific journals. It's really difficult to achieve that. You need to have really excellent drawing skills and how you make nice figures. A website. If you would like to have more information about our group, you can go to this link and find more information about our team and the work we're doing. I was invited to U.S. Congress. I could never imagine that I would have this in my life. And that was a great honor for somebody who became American citizen only 7 years ago. And that was a great experience also for me. I was invited there to present to members of the Congress the research we are doing here at Cook and the vision I have about epilepsy research at the national level. Funding. We know money is important. So we brought here $6.7 million so far in terms of funding, federal and foundation funding for supporting our team, and we'll keep trying to bring more. Now it's more tough. Okay, so that's it for today. For what has been done so far, I would like to say a few words about the future. So Cook Children's healthcare system provides great support, and through this support, we are able now to expand our team through a 5-year strategic plan expansion. So we built 3 now new teams, in addition to the existing teams on epilepsy research, movement disorders and stroke, and mental health. And within the next year or 2 years, we'll start hiring all these people. So all this work could not have been done with my team. And I know I don't say positive comments frequently, but when I say I mean it to my team. They work really hard. There are members in the team that I text them 10 p.m. in the night and they respond. And then I want this slide by tomorrow. And then they do it. And I would like to say that thank you. You're an amazing team. I have the privilege to work with all these amazing people. All of you have great future as researchers. Some of them, they escape the team, but I will try to bring them back. Because I believe that all these smart people, they will be able to do really great things in future, and we need to build on new generations. So I would like to close my talk with thank you so much, of course, the Cook Children's Health Care System with all the support, Dr. Stigal, for all the support he provides. But most important, I would like to thank the parents and the families who participate in our studies, because whatever we do it for them. And then we care about the kids, and all the research we do is focusing on how these children, they will become healthy one day in life. Thank you so much.
If you have questions, please come to the mic. You probably should go soon because I have 8,000 questions.
That was an excellent talk. Thank you so much. I'm Lane Lanier. I'm from the cardiac ICU here at Cook. I can't help but overlay so many things that are going on. And as a lot of people know, cyanotic heart disease, there are changes going on to the brain in utero. And then as we do surgery on these babies, they're undergoing insults also to their brain with deep hypothermic circulatory arrest and now selective integrated cerebral perfusion during longer bypass times. It's not infrequent that we miss seizure activity in the immediate postoperative period. It is difficult in patients who are so sick from hemodynamic standpoints to take this wonderful technology and bring it to the bedside in the NICU. But from your talk, I'm seeing some of that happen. What limitations or what can we expect in terms of innovation with MEG and the types of EEG that you're doing to bring it out of maybe one area of the hospital and move it to the bedside? Because I see so much opportunity even within my area for some of these technologies. Could you comment on that?
Yeah, excellent comment. Thank you for this question. The technology is there, and that's the impressive thing. We have so many tools, but these tools are not used. And they're not used because, and I always say that to my friends in UTA in bioengineering department, because like clinicians and then bioengineers, they talk different languages, like it's like Greek and English or something. And then we need to translate all this work that is done in the technology level into the clinical practice. When I was in Boston Children's Hospital and I was doing some work in the NICU by myself, I was really involved with it, like, and I'm not the ideal guy to go walk around in the NICU, like, and I was working with these kids and collecting evoked responses. And then I asked the neonatologists there, like, how you make an assessment of the neurological status of the infants? And then they told me, we clap. For assessing the nutritive suckings, the nurses put the finger in the mouth to see if the infant is sucking properly. EEG, they do some EEG, but like there's no localization, there's no evoked responses. And then I realized and that's like, okay, Boston Children's Hospital is a big hospital. It's like, if they don't do that here, where do they do that? And then I realized there's so many opportunities that can be done. And I fully agree with you. Like, we need to do more work on all working together. And we have the technology, but and from you, the clinicians, like, we need the questions. Like, tell us, like, okay, that's a great idea. And then we have this discussion with the clinical team, with Scott Perry, with Warren Marks, like, oh, Christos, that's the problem. It's not what you're focused. Because engineers, we focus on our tools. Like, we go on our tools. We have our tools and we work with that. But we need the clinicians to tell us what is critical. So I will be more than happy to discuss more about like ideas about the PQ, the NICU, like, and projects. But the technology is there. That's my comment.
Thank you so much, Christos. Just fantastic. Will you speak a little bit about the unique setup? You said here that you said we're one of the only ones doing at the same time, doing MEG and HD-EEG. Will you walk through, like, how many centers, everyone's got a functional MRI, how many centers have an HD-EEG, how many centers have a MEG, how many centers have a TMS, and then who's doing what with combinations of those things? Mm-hmm.
So the MEG technique is like not so much used, like, although it has been around for so long time. And I think this is mostly because like we don't have enough users or people who know how the MEG works. And there's need work to be done there on how to educate people to do MEG properly. It's to give you a number, I think United States there's no more like 15 or 20 place maximum that they have a MEG. The high density G is a quite interesting technique. It has been in the cognitive or cognition field for a long time, but in epilepsy it's something totally new. They don't have any CPT codes like to charge for that. And then I was giving a talk in Baltimore in the American Clinical Neurophysiology Society meeting last week, and then there was two sessions, one for the MEG community and one for the EEG community. When I was presenting to the MEG community about the EEG, they said like, oh, you're in the wrong room. So like, no, we should talk to each other. They're all techniques. So we are one of the very, very few places, like maybe in the world, that we use this combined MEG high density G. This offers like significant information about complementary information. What I saw, like, because like sometimes you see spikes on the MEG, but not on the EEG, and vice versa. And we don't know exactly why. And that's why we do work with my team to see this kind of orientation of the sources. The MRI scans, they're all over the place. They're in many places. But it's also like how you work with that and how you integrate that to the research questions.
And what about TMS?
TMS it's a very interesting method. It's used for brain mapping of motor activity. The language is not so easy. There's also studies for doing language mapping. We tried that in some kids, like it didn't work quite well. There are places that do like extensive work. There are places they don't work at all with TMS, and then with just the way the TMS is sitting here. Actually, when I came here and I was interviewed for the position, I saw the TMS sitting in the corner. There was a bucket on the top of that. And it's like, wow, we have a TMS here sitting on the corner. Like in Boston Children's, like we were begging to have a TMS or access to TMS. It's like, oh, yeah, we have that here. It's like, okay, we need to use that. So it's the tools that they're available, the tools, but also like how you use the tool, and then you have the proper people to train them and keep the people, have the machines up and running.
Lane, I know you have more questions. Someone needs to get behind me. Oh, go ahead. I literally have 5 questions. I'm gonna keep asking.
Hi, good morning. Thank you so much for presenting. This is really, being new to Cook Children's within the last 18 months and coming from an academic center, this is really impressive to see, so thank you. Clinically, I practice as a pediatric hospitalist here at Cook Children's, and so I'm curious about your thoughts. You know, so much of this technology being applied in very specific settings for neurologic disease. As a hospitalist, along with my neurology colleagues who are so patient with us when we consult them, we see a lot of patients of new parents of babies who are worried about abnormal movements that they think are seizures. Overwhelmingly, most of them are not. Do you foresee a time where certain technology that you're looking at or that is similar to what you're doing could be applied in an urgent care or pediatric office setting to avoid unnecessary hospitalization for abnormally reported movements? Is there something that you foresee in the future could be applied in those settings? Because those parents come, the experience is tough. I have some funded time as a physician director in patient experience as well, and so to avoid hospitalization for a brand new baby where parents are tired and stressed and there's so much going on, avoid unnecessary studies like EEGs that we routinely order all the time, or MRIs even, or referrals to neurologists that don't need to be done. Curious about your thoughts about those patients.
So it's not work I'm doing, or so it's not work I'm doing, but I know that there are other places, like they have these cameras they put in different places. They do analysis of the videos about abnormal movements, and there's some work even for cerebral palsy. And there's definitely technology there. When you have somebody in a freely space, like to move hands and things like that, I know how the technology will work quite well, but definitely this is something that it's work that can be done. I have heard places doing this kind of work for movement disorders and mostly for cerebral palsy work, for early detection. Yeah, if you know something, like I would like to hear more, but it's not something I'm doing or working on. Tia told me we have 5 minutes, Tia? Yeah.
As I'm looking at the pictures of the MEG, I'm watching the size of the patient going in seems to be older, but for me, outside of the infant range. Can you talk about the limitations in terms of size with the MEG and I guess in terms of the patients you're doing, because I'm seeing more of 5, 6, 7-year-olds. I'm not seeing infants. Talk about that if you don't mind, please.
Sure. I know this topic quite well. I spent 4 years of my life building what's called Baby MEG in Boston Children's. Boston Children's, they spend millions of dollars for building a MEG specifically tailored for toddlers. And this is where the kind of business comes in line, or you should discuss with business people the research, because the hospital built this system. I built it actually by myself with one of my ex-mentors. And then after a while, we realized that kids who go into epilepsy surgery in this early age, no more than 10. So they spend all this money for a system that could be used in few kids. And then the hospital decided we need to do something about that. So let's do normal development studies. And then we're bringing healthy toddlers for measuring them. And as a parent, I should know that, but a toddler, he never likes laying down on that. He would like to move all the time. So yeah, there's technology, but we need to, and then there's room to be done, a lot of work to be done there. That's why the high density also is more easy for this population. But yeah, there's need to discussion about business and clinical need and research. Scott.
Good morning. Fascinating talk.
Thank you.
Whoever brought you here, complete genius. Complete freaking genius.
You brought me here.
Oh, okay. Oh, yeah. Yeah, why don't we, if you could, sum up for the folks what it was about Cook Children's, what it was about the pitch that made you commit academic suicide and come here and be successful.
Frankly speaking?
Yeah.
You're a very convincing guy. Yeah, because I remember our exchange emails. I told you, because I was Boston Children's, and then I went to my chief there, very good friend, and she gave me great support. And I told her, I have this offer from Cook Children's. And I still remember her reaction, Cook Children's? What is that? And she told me, she told me, Christos, this is a professional suicide. And I was coming back home, I was saying to my ex-wife now, like, I was telling her, like, are we sure what we're doing? And then I wrote an email to Scott. I was like, okay, I will ask him this and this and this and this and this. And Scott, I was in Greece during this time for a trip. And then you respond like, yes, 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 yes. It's like, bingo, this guy, he knows what you're talking about. So you brought me here.
Well, I was thinking the answer was more, the fact was, what was great about it is that we don't have everybody doing research, right?
Yes.
The opportunity is the patients are here, they are ready,
Yes.
they need the care, they need the treatment, and we have the resources to do it, we have the patients to do
That's-
it, and we have the time
Yes.
to do it. And you know, as we talked, I said, hey, if the MEG's not being used, bro, it's all yours. Go for
Yes.
it. Like,
No,
gonna charge
yeah.
you for it.
To your point, that's an excellent point. During the interview here, I realized the opportunities. To be honest, like so, I remember our tour in the MEG, and you told me, oh, this tool like is not used so much. Like wow, and then I saw the TMS. Nobody used the TMS. Like the TMS is mine. He told me, yeah, it's yours. Like wow. And then I told him, I told you like I wanted high density G. He told me, yeah, we'll buy you one. It's like, okay. So then that's unique. And then exactly what you said, like I realized that that's a field, and then I can just grow up fast in this field with passion and with hard work. Yeah.
Just fantastic. We're going to keep ourselves on time. Thank you so much, Christos.
Okay, thank you.
Thank you so much.
Thank you.